This is a GCSE chemistry tutorial focusing in on topic 7, which is the organic chemistry unit. This video will focus on biochemistry, which is a triple only topic. In this tutorial video we will look at 3 different naturally occurring polymers. We'll have a look at the key properties of DNA, and we'll have a look at the formation of both polysaccharides as well as polypeptides. During your biology GCSE, you should have come across a number of naturally occurring polymers. For chemistry, you need to know about 3. These are DNA, polysaccharides, and polypeptides. First, we will look at polysaccharides. These have to do with carbohydrates. Carbohydrates are made up of molecules containing carbon, hydrogen, and oxygen. They have the general formula of CxH2OX and are made up of at least one type of sugar molecule, which leads us to what is the most common simple sugar. The most common simple sugar is glucose, C6H12O6. Glucose is by far and away your body's preferred fuel source. All other sugars are converted into glucose within your body in order to then carry out respiration. Glucose is an example of a monosaccharide. It is a simple sugar. This means it's made up of only one sugar unit. Other monosaccharides include fructose and lactose. Sucrose, on the other hand, is a far more complicated molecule. It is an example of a polysaccharide, which means it's made up of multiple monosaccharides. Sucrose is made up of glucose and fructose. It's formed in a condensation reaction, meaning it is a condensation polymer. We looked at condensation polymers in the last tutorial video. Glucose can also form very, very large polysaccharides. These include starch, which can be broken down into glucose using amylase. It can form glycogen in terms of being a storage. You will have looked at this in biology in terms of insulin. And finally, it can form cellulose, which is a fibre and can be found in plant cell walls. We also need to look at proteins. Proteins, as you should know from biology, are long polymer chains made up of amino acids. Amino acids have a carboxylic group and an amine, that's an NH2 group, so can join together in a condensation reaction. In the last tutorial video, we looked at condensation reactions where each molecule, each monomer, had 2 of the same functional group. For polypeptides, the amino acids which form the monomers have 2 different functional groups attached onto the one monomer. The polymers that are formed are known as polypeptides. These polypeptides then form up to form proteins as they fold in the correct manner. In this reaction, we have 2 amino acids. For this example, we have got 2 of the same amino acids, which is glycine, which is the smallest of the amino acids. In order to react, the OH part of our carboxylic group reacts with 1 hydrogen of the amine group, causing the release of 1 molecule of H2O. In doing so, the amine group, the NH2, and the carboxylic group, the COOH, join together to form CONH. This then causes the release of this molecule of H2O, and in doing so causes the formation of a peptide bond. We can then write this as a polymer by putting brackets around it, making sure, as we did before, that the bonds cross through the brackets, and putting an N. The amount of water we produce, so our NH2O, is the same as the number of polymer links that have been made. Polypeptides are known as proteins. They have lots of uses in the human body, for example as catalysts in the form of enzymes. They could be hemoglobin transporting oxygen, they could be antibodies, or they could be any number of other parts of our body. Polypeptides and proteins can contain different amino acids in their polymer chains, and the order of the amino acid is what gives proteins their different properties and shapes. Finally, we have DNA. DNA is deoxyribonucleic acid, and you will have come across it in biology. DNA is one of the most important molecules in the human body, or in most organisms. It contains genetic instructions that allow the organism to develop and operate. It's a large molecule and has a double helical structure. Which we can see here. We will look at this structure in more detail on the next slide. Here we can see the base pairing that is going on in the helical structure in more detail. DNA is made of two large polymer chains of monomers called nucleotides. So our nucleotide here is our monomer. Running down all of each side, we have our two long polymers. In the nucleotide, we have a phosphate and a sugar, making our sugar phosphate backbone, and we have our nitrogenous base. The base can be one of four options. These are adenine, A, thymine, T, cytosine, C, and guanine, G. For GCSE chemistry, you only need to know these by their initials. These bases on the different polymer chains pair up with each other and form crosslinks, known as hydrogen bonds, which hold each base pair together. This gives it its double helical structure as the pairs line up. The order of these bases then acts as a code for the organism's genes and therefore controls what the genes are that are going to be made, as well as being affected by any mutations that may occur. You'll look at DNA in far more detail in GCSE biology.